For centuries, the ancestry of Black Americans was hidden behind a single word, African. That word described millions of people whose ancestors actually came from dozens of societies, languages, kingdoms, and regions spread across one of the most genetically diverse continents on Earth. Modern genetics is changing that picture. When scientists compare African American genomes with European genomes and with DNA from populations across Africa, they do not find one simple genetic identity. They find a record of migration, forced movement, admixture, isolation, survival, and biological inheritance. On average, African Americans carry a majority of sub-Saharan African ancestry together with a substantial European component, but individual genomes vary dramatically. The African portion itself is not one thing. It contains signals connected to multiple regions of West and Central Africa, while the European component carries its own history. The result is not merely a comparison between Black and European DNA. It is a record of two continents becoming intertwined through one of the largest forced migrations in human history, while ancient genetic differences shaped by climate, disease, and population movement remained embedded underneath the modern population. The first discovery scientists had to make was that Black American is not a genetic category in the same way ancestry is a genetic category. It is a social, cultural, and historical identity containing enormous biological diversity. Two people who both identify as Black American can have very different ancestral profiles, while two people assigned to different racial categories can share substantial ancestry. Genome-wide studies have repeatedly demonstrated this variation. African ancestry in African Americans commonly falls around 3/4 of the genome on average, while European ancestry is often around 1/5, but these are population averages rather than rules for individuals. Some people have far more African ancestry, some more European ancestry, and some carry small contributions from other populations as well. The important point is that the genome does not recognize the social boundaries that societies created. It records inheritance. That distinction becomes especially important in the United States because racial classifications were historically created through laws and social systems rather than through precise biological definitions. A person could be classified as Black regardless of how much European ancestry they carried, and another individual could have African ancestry without being classified in the same way. Genetics exposes the complexity underneath those labels. The African side of this story is extraordinary because Africa contains the greatest overall human genetic diversity on Earth. Our species originated there, and populations across the continent have been structured and evolving for hundreds of thousands of years. When relatively small populations left Africa, they carried only a subset of the diversity that remained on the continent. This founder effect helps explain why non-African populations generally contain less genetic diversity than Africa as a whole. But Africa itself is not one genetic population. A person from Senegal has a different ancestral history from someone from Nigeria, Ethiopia, Cameroon, Congo, Namibia, or South Africa. Language, geography, climate, migration, and population size all shape those differences. That matters enormously for African American ancestry because the African portion of a Black American genome can reflect several African regions at once. Historical records of the transatlantic slave trade show major source regions including Senegambia, Sierra Leone, the Gold Coast, the Bight of Benin, the Bight of Biafra, and West Central Africa. Modern genetic studies broadly recover signals corresponding to these regions. The genome cannot restore every lost ethnic identity, but it can provide statistical clues about where ancestors may have lived. That ability is especially important because slavery deliberately disrupted ancestral identity. Enslaved Africans were often recorded without the names, languages, family connections, or ethnic information that modern descendants would need to reconstruct their origins. A person might appear in an American record only as an enslaved man or woman with no reliable indication of whether their family came from Yoruba, Igbo, Wolof, Mandinka, Akan, Menda, Congo, or another population. Genetics cannot magically recover every forgotten name, but it can compare segments of African American genomes with reference populations in Africa and identify which regions are statistically closest. Those matches are not perfect ethnic labels, because present-day populations are not identical to historical populations. Borders changed and communities mixed over centuries, but they can narrow the possibilities. A single African American genome can contain ancestry connected to several parts of Africa. Because enslaved people from different regions were brought into the same colonies, and their descendants eventually formed new communities. This means DNA preserves both the fragmentation of African identities and the creation of new identities in the Americas. For families whose written genealogies stopped during slavery, that information can be deeply significant. It cannot recover everything, but it can provide a geographical starting point that did not exist before genetic testing became possible. Ancient DNA is making that recovery even more powerful. Researchers have begun sequencing remains from historical African American cemeteries and comparing them with modern genetic databases. One important example is the Catoctin Furnace site in Maryland, where African American laborers lived and worked between the late 18th and 19th centuries. Researchers analyzed genomes from historical individuals and compared them with large databases of living people who had consented to genetic research. They identified tens of thousands of genetic relatives among modern participants. This created something that written records often could not provide: a direct molecular connection between people who lived centuries ago and people living today. The study also found African ancestry associated with West and Central African populations, together with European ancestry, consistent with historical connections to Britain and Ireland. The importance of this goes far beyond a scientific percentage. It demonstrates that an archaeological burial can become part of a living family tree. A person who died in 19th century Maryland can still have descendants carrying pieces of that person's genome. For descendants of enslaved people, this offers another path into a history that paper documents often left incomplete. DNA cannot replace genealogy, archaeology, or oral history, but it can connect them. A cemetery can become a genetic bridge between generations. Now comes one of the most disturbing findings in the comparison between Black American and European DNA. The European ancestry inside African American genomes is strongly sex-biased. Genetic studies show that European contributions were disproportionately inherited through male ancestors, while African ancestry was disproportionately represented through female ancestors. This pattern is consistent with the historical structure of slavery, in which European men possessed overwhelming legal, economic, and physical power, while enslaved African women were especially vulnerable to sexual exploitation and coercion. The genome cannot tell us the circumstances of every individual conception, and it would be wrong to assume that every European ancestor represented an act of violence. But at the population level, the asymmetry is unmistakable. European ancestry did not enter African American genomes through a balanced process in which African women and European men contributed equally by choice. The proportions reflect the social structure of the slave system and later American history. This makes the DNA an unusual historical record. It does not preserve emotions or names, but it preserves demographic patterns. A child born from a coerced relationship inherited DNA from both populations. That child had children. Those children had descendants. After hundreds of years, the original events became invisible to memory but remained visible in the genome. The biology, therefore, carries evidence of a historical system that was designed to control bodies and families. The timing of that European admixture provides another important clue. Population genetic studies indicate that much of the major African-European mixing that shaped African-American ancestry occurred during slavery and the generations around emancipation, especially in the southern United States. This does not mean every family followed the same pattern. Migration, intermarriage, and additional immigration continued after emancipation. The Great Migration later moved millions of Black Americans from the rural South to northern, Midwestern, and Western cities. Caribbean and African immigration also added new ancestry to American Black populations, but the ancestral foundation of African Americans in the United States was established during the period when captive Africans were forcibly brought to North America, and their descendants lived under slavery. That historical period, therefore, remains visible in modern DNA. Genetic models can estimate when large-scale admixture occurred by looking at the length and distribution of ancestry segments across chromosomes. Longer segments generally point toward more recent mixing, while shorter fragments reflect older events. This allows scientists to distinguish historical periods. The genome cannot name the plantation or the exact year, but it can provide a timeline consistent with the documentary evidence. That is one reason genetics is such a powerful complement to history. A shipping record can show that Africans arrived. A family document can show that a person lived somewhere. DNA can reveal how those movements change the ancestry of later generations. The European side of the comparison has its own ancient complexity. Modern European populations are not genetically pure descendants of a single ancient European group. Ancient DNA has shown that Europe was shaped by several major ancestral streams, including hunter-gatherers, early farmers related to populations from Anatolia and the Near East, and steppe-associated populations that expanded during the Bronze Age. Later migrations added still more layers. This matters because the European ancestry inside African American genomes does not represent one single European gene pool. It contains whatever European populations contributed to African American ancestry over the past several centuries. Some segments may reflect ancestry connected to the British Isles. Others may trace to continental Europe or later American admixture. The genome is therefore a mosaic on both sides. African ancestry is a mosaic of African populations, and European ancestry is a mosaic of European populations. This is one reason researchers increasingly prefer terms such as ancestry rather than treating racial categories as fixed biological units. A person can have ancestry connected to multiple continents without belonging biologically to separate categories. The genome is a record of movement. It is not a collection of sealed boxes. The deepest difference between African and European genetic histories is the amount and structure of genetic diversity. Because Africa contains the longest continuous history of modern humans, populations there accumulated extensive genetic variation. Researchers sequencing southern African populations have found millions of variants that were absent from older global data sets. One major study identified approximately 3 million previously unreported variants in African genomes, while more recent sequencing of cosine groups revealed more than 1 million additional single nucleotide variants not previously cataloged. These discoveries matter because the global reference genome historically represented European populations far more heavily than African populations. If the reference is incomplete, medical and ancestry models are incomplete too. African American genomes inherit only a portion of Africa's enormous diversity, but some variants that are rare in Europe remain important in people with African ancestry. This has direct consequences for medicine. A genetic variant that is uncommon in European datasets may be common enough in African ancestry populations to influence disease risk or drug response. If scientists look only at European genomes, they can miss those signals. Expanding African genomic datasets, therefore, benefits not only Africans and African Americans, but everyone, because comparative genetics improves the accuracy with which researchers identify biological mechanisms. One of the clearest examples of this ancient African adaptation is the sickle-cell variant in the HBB gene. When a person inherits one copy of the sickle allele, they have sickle cell trait rather than sickle cell disease. When two copies are inherited, sickle cell disease can occur and can cause severe complications. Why would a variant capable of causing such a serious condition become common in some populations? The answer lies in malaria. In regions where malaria was historically common, people carrying one copy of the sickle allele had partial protection against severe malaria. Those individuals were more likely to survive and reproduce than people without the allele in environments where malaria pressure was intense. Over many generations, natural selection increased the frequency of the variant. This is one of the clearest examples of balancing selection in humans. The same genetic change can be beneficial in one form and harmful in another. For African Americans, sickle cell ancestry provides a direct connection to environmental conditions faced by ancestors in malaria-endemic regions of Africa. But it is essential to avoid turning this into a statement about race. The relevant biological factor is ancestry from populations historically exposed to malaria, not skin color. Similar sickle variants also occur in other populations with deep malaria exposure. The genetics reveals an evolutionary history, not a racial hierarchy. APOL1 provides another example of ancient selection with modern consequences. The APOL1 G1 and G2 variants became more common in parts of sub-Saharan Africa because they improved resistance to certain strains of trypanosoma parasites associated with African sleeping sickness. But inheriting two high-risk copies can increase vulnerability to certain forms of kidney disease. This is another evolutionary trade-off. A variant that helped ancestors survive infectious disease can increase disease risk in a different environment. The presence of APOL1 risk variants is more common among people with substantial African ancestry, including many African Americans. Researchers have therefore been studying how these variants affect kidney health and how genetic testing could eventually improve treatment. But again, APOL1 is not a black disease gene. Like carrying a high-risk genotype does not guarantee kidney disease, and environmental, genetic, and clinical factors all influence outcomes. The lesson is that ancestry can change the frequency of important variants, but individual biology is always more complicated than population averages. This is precisely why researchers need diverse datasets. European-dominated genetic databases might not capture African-specific variation adequately. African American genomic studies are helping correct that imbalance by identifying variants that matter for populations with African ancestry. The immune system reveals another difference. Researchers have found population-level differences in immune gene expression and inflammatory responses between people with African and European ancestry. A well-known line of research has suggested that African ancestry is associated with stronger inflammatory responses to some infectious challenges. Such differences can make biological sense because African populations historically encountered a wide range of pathogens including malaria, trypanosomes, and numerous bacterial and viral infections. Natural selection can favor immune variants that improve pathogen defense. But stronger inflammation is not automatically better. The same inflammatory pathways that help fight infection can increase vulnerability to autoimmune or inflammatory disease. European populations have their own adaptations to historical pathogens, and those variants can produce different immune profiles. So the correct conclusion is not that one population has a stronger immune system. It is that populations can carry different immune architectures because their ancestors faced different environments. This distinction is essential. A person's actual immune response depends on genetics, age, environment, infection history, nutrition, and many other factors. Race cannot tell a doctor exactly how an individual's immune system behaves. Genetic analysis can help, but only when the relevant variants are measured directly. European ancestry adds another deep layer through ancient Neanderthal admixture. Most people whose ancestry is largely outside Africa carry around 1 to 2% Neanderthal-derived DNA because modern human ancestors encountered Neanderthals after leaving Africa. These genetic contributions can influence immune function, metabolism, skin biology, and other traits. Some African populations also contain small amounts of Neanderthal-related ancestry because later populations with Eurasian ancestry moved back into Africa. This means even the distinction between African and European ancestry becomes less absolute when we look deep into evolutionary time. Human groups repeatedly encountered one another, and genes sometimes crossed boundaries that populations otherwise maintained. A modern African-American genome with European ancestry can therefore carry a small portion of an ancient Eurasian history that goes back tens of thousands of years. But that ancient component does not make African Americans less African or more European. It simply demonstrates how connected human evolutionary history has always been. The same genome can contain segments associated with multiple ancient populations. Some arrived recently, some are tens of thousands of years old. Others may have been common to humanity long before modern population categories existed. The transatlantic slave trade created another remarkable genetic event by bringing people from populations that were often genetically distinct into the same American environment. In Africa, Yoruba-related Igbo, Wolof, Mandinka, Akan, Mende, Congo, and many other populations had their own languages and histories. In the Americas, enslaved people from several source regions were placed together, and their descendants formed new communities. Over generations, previously separate African populations became genetically mixed. Modern African Americans, therefore, carry combinations of ancestry that may have been rare or non-existent before the Atlantic slave trade. This is one reason a single person can have ancestry pointing toward several regions. The mixing occurred across generations, not necessarily in one event. An ancestor from West Africa could have children with descendants of people from Central Africa, and those descendants could later have children with someone whose ancestry was mixed again. The genome accumulates those layers. The result is not one African identity, but an American population carrying many African histories simultaneously. This makes African American genetics scientifically unique. It represents both the destruction of specific ancestral structures and the creation of new population structures in America. The genome therefore documents an enormous historical transformation. It does not simply tell us where people came from. It tells us what happened when those populations were forced together. This history also explains why African American ancestry cannot be reconstructed by comparing people only with one modern African population. Present-day African populations have their own recent demographic histories. The transatlantic slave trade began centuries ago, so the best reference for a 19th century African ancestor is not necessarily a modern population living in the exact same location. Researchers therefore use many African data sets and statistical models to estimate the most likely ancestral sources. As sequencing expands, these models become more detailed. Future databases may identify finer geographic patterns that commercial tests currently cannot resolve. A person might eventually be able to see that one part of their ancestry is strongly associated with Senegambia, another with the Gulf of Guinea, and another with West Central Africa. Such conclusions will remain probabilistic, but they can still transform family history research. For descendants of enslaved people, the ability to recover ancestral geography can provide a connection to specific regions and communities that written documents erased. It is not the same as discovering a great-great-grandparent's name, but it can create a historical bridge. That bridge becomes even more powerful when ancient DNA and modern relatives are combined. The Catoctin Furnace study showed that historical African American remains could be genetically connected to living people. This demonstrates a future in which archaeology and genealogy become deeply interconnected. A burial site can reveal not just who lived there in the past, but where their descendants are today. A family may discover that a person buried 200 years ago shared genetic segments with living relatives. Researchers can then combine those relationships with historical documents to reconstruct migration routes. This approach has immense potential for African American history because traditional genealogy often encounters a barrier at slavery. Records before emancipation can be incomplete, inconsistent, or deliberately dehumanizing. DNA offers an independent source of information. It does not replace the names and stories that make genealogy meaningful, but it can identify relationships where paper records fail. The result is a new kind of historical evidence, one that allows the descendants of enslaved people to reconnect with biological relatives who existed before the last names and legal documents commonly used today were available. Now comes the central question. What exactly shocked scientists when Black American DNA was compared with European DNA? The most surprising result was not one exotic mutation or a secret ancestry group. It was the sheer complexity of the comparison. African Americans carry substantial African ancestry, but that African ancestry itself can be connected to multiple populations. They also carry a significant European component that is strongly sex-biased toward male ancestors. Their genomes therefore preserve the demographic structure of slavery and later American history. At the same time, African ancestry carries deep genetic diversity and variance shaped by ancient environments. Some of those variants influence modern health. European ancestry brings a different evolutionary history, including ancient European population mixtures and, in many cases, Neanderthal-derived segments. The two populations are therefore not separate biological types. They are overlapping human populations with different histories. When scientists compare them, they see divergence at the level of allele frequencies and ancestry composition, but they also see enormous shared biological structure. This is why the idea of a black gene or European gene is usually misleading. Individual variants can have very different frequencies in different populations, but few are exclusive to one broad racial category. Most genetic variation is shared across populations, and many differences involve frequencies rather than complete presence or absence. A variant can be common in African populations and rare in Europe, while still existing in some Europeans. Another can be common in Europe and rare in Africa while still appearing in African populations. The boundaries overlap. This is why genome-wide analysis is needed. Researchers examine thousands or millions of genetic markers together to estimate ancestry. One marker rarely tells the story. Thousands of markers create a statistical pattern. Modern genetics, therefore, does not reveal fixed biological races. It reveals population structure. The structure is real, but it is flexible. It changes when populations migrate, mix, grow, or shrink. The medical implications are enormous. African American populations are underrepresented in many genetic datasets compared with their proportion of the global population. This can reduce the accuracy of polygenic risk scores, drug response models, and disease prediction tools for people with African ancestry. African-specific variants can be missed if the reference data are dominated by Europeans. APOL1 is one example. Sickle-cell variation is another. Other pharmacogenetic variants are distributed at different frequencies across ancestral groups. Researchers are therefore increasingly designing studies that include African American participants and broader African populations. The goal is not to create different standards of medicine based on race. The goal is to create models that understand individual biology more accurately. Ideally, a doctor should use a person's genome, medical history, family history, and environment rather than assuming that every Black patient has the same biology. Population data can help identify which genes matter, but the future is individualized. African American genetics is helping move medicine toward that future by making previously invisible genetic variation visible. There is also a historical significance to the African diversity found in Black American DNA. The transatlantic slave trade often treated humans as commodities, reducing complex individuals to numbers on manifests and ledgers. Modern genetics reverses that logic. Instead of seeing an anonymous shipment, researchers can see populations. Instead of a generic African label, they can sometimes identify regional ancestry. Instead of a person disappearing into a plantation ledger, their descendants may be genetically identifiable centuries later. DNA cannot repair the moral damage of slavery, but it can challenge the historical erasure that accompanied it. It reminds us that enslaved people came from societies with languages, political systems, artistic traditions, religions, technologies, and families. Their descendants still carry portions of that history. The genome cannot tell the complete story of a Yoruba ancestor or a Congo ancestor, but it can establish that the ancestry exists. It turns it, somewhere in Africa, into a much more specific scientific question. And as databases improve, that question becomes more answerable. One of the most important conclusions, however, is that African American DNA does not represent an unchanged African past. The population formed in America and evolved in America. Its members experienced new diets, diseases, environments, migrations, and social conditions. European ancestry entered the population. Native ancestry entered some lines. Later African and Caribbean immigration introduced additional diversity. Natural selection continued. Genetic drift continued. The genome therefore reflects both the ancestral environment and the American environment. This is why it would be wrong to describe African Americans simply as Africans who were transported to America. Their ancestors were Africans, but their population became a new population in the Americas. It developed its own genetic structure, cultural traditions, and history. The genome contains evidence of that transformation. It is one of the clearest examples of how migration can create a new population within only a few centuries. The same principle applies to European Americans. Their genomes are not simply European genomes placed in America. Different European populations migrated to the United States, then mixed with one another and with non-European populations. Selection and drift continued. New migrants arrived. The population changed. When scientists compare European Americans with African Americans, they are comparing two populations formed partly through the same country, but with radically different historical origins. The comparison, therefore, contains the imprint of American history itself. The genetic distance between the groups is partly ancient, partly continental, and partly the product of events after Europeans and Africans arrived in the Americas. This makes the comparison scientifically fascinating. It is a population genetic record of colonization, slavery, migration, and later demographic change. The final lesson may be the most important one. DNA can preserve evidence, but DNA cannot tell us what that evidence means morally. A genome can show that European ancestry entered African American populations disproportionately through male lines. It cannot tell us how an individual ancestor experienced that relationship. It can show African ancestry connected to certain regions. It cannot tell us the full culture of the ancestor who carried it. It can reveal a disease-associated variant. It cannot decide how an individual should respond to that information. Genetics is powerful because it measures biology. History, anthropology, genealogy, and lived experience are needed to interpret that biology responsibly. When scientists compare Black American DNA with European DNA, the result is not a story about two separate races. It is a story about ancestry under pressure. African populations contributed enormous genetic diversity. European populations contributed another layer. Slavery forced these histories together. Generations of families carried the mixture forward. Natural selection preserved some variants. Demographic bottlenecks changed others. Migration reshaped the population again. Today, researchers can reconstruct parts of that history from millions of DNA markers. You are carrying migrations, you are carrying adaptations, you are carrying ancient survival strategies. You may carry ancestry from several African regions at once. You may carry European ancestry introduced hundreds of years ago. You may carry genetic variants shaped by diseases your ancestors never saw directly but survived through. And somewhere inside those chromosomes is information about people whose names were lost. The genome is remembered in its own language. It remembers through patterns. It remembers through inherited segments. It remembers through variants passed from parent to child. And sometimes it remembers what history tried to erase. That is the real shock in the comparison between Black American and European DNA. Not that one population is superior, not that one is completely separate, not that one possesses a secret genetic code. The surprise is that the genome can reconstruct a historical journey that written records could not fully preserve. It reveals Africa's enormous diversity. It reveals Europe's contribution. It reveals the unequal structure of slavery. It reveals ancient adaptations to malaria and parasites. It reveals the genetic consequences of migration and forced mixture. And it reveals that the descendants of enslaved Africans still carry biological links to the continent from which their ancestors were taken. The history was not destroyed. It was fragmented. It was scattered. It was hidden. But much of it remained inside the cells. And now science is learning how to read it. That is why the most important comparison is not Black versus European. It is past versus present. The people who were separated by ships are connected again through DNA. The families whose records were broken can sometimes be connected through shared ancestry. The communities whose histories were reduced to numbers can sometimes be identified through their genetic signatures. And the ancient environments that shaped human survival can still be seen in variants carried by people alive today. The genome does not erase history. It confirms that history happened. And it proves that the people who survived it carried their ancestors forward.